So the next branch is actually a bigger branch of the immune system. This is what we call our humoral response. Now it comes from the word humor, which in ancient times meant bodily fluids. Okay, so the ancient Greeks used to talk about the humor, and that is the bodily fluid. So the cell-mediated response has to do with infected cells, and the humoral response sorry, I got cut off there. The humoral response has to do with infected bodily fluids. So this is the area surrounding the cells. But before we do that, let's just have a really quick recap on the cell-mediated response. So here's the cell-mediated response. You have an infected cell. It gets engulfed by a macrophage or a phagocyte. They present it on their MHC2s. They activate a T helper cell. After they activate the T helper cell, they undergo mitosis and they become a whole bunch of, oh, that's not a very nice way of writing that. They become all of these cells down here, cytotoxic T cells. So this is a T helper cell, cytotoxic T cell. What they do is they have cytokines and they also have perforins and other proteins, and they end up killing the cells that way. So they will kill off the infected cells by poking holes in them. So I want you to stop. You can have a go at these if you want to, but I'm going to move on and start to talk about our last area. So we've done our physical and chemical barriers and microbiological. We've done the innate immune response. We've done the cell-mediated response, which has to do with the T cells. Now, the final one that we've got to talk about are the B cells and the humoral response. Now, it's going to take up more than one video. We are on page 14, and let's talk through our story. So to begin with, I'm going to ignore this stage here because it's a bit more detail than what you need to talk about. So what we get is we get a T helper cell again. So the thing that we're missing from here is the APC presents, oh, sorry girls, you seem to have lost me there. I'm just going to get this back. I'm going to pause and we'll figure this. Okay, magic, it's back again. So I want you to have a look at this and you can pause if you need to and change the annotations. But I don't like that first diagram, but I like the rest. So that's why it's there, and I didn't block that one out for you. So the thing is, it's the same start as in the cell-mediated. This is the same as cell-mediated. And when we put them all together in a nice little summary bit at the end, you'll see how we can more easily remember what's happening. So what happens is the antigen presenting cell, so this could be a macrophage, it could be a dendritic cell, any one of those from the innate immune system. The macrophage APC presents its antigen to a naive B cell or just a B cell, and they get activated. Oh, sorry, presents its antigen to a T helper cell, and then the T helper cell activates a B cell. Now, what the B cell does is it divides to make two different types of cells. So this is the same as the T cells. They activated to make cytotoxic T cells, and they did make those T memory cells, but technically they can't ask you about T memory cells on the exam. They can ask you, however, about B memory cells. So this is a B plasma cell, and this is a B memory cell. So once you've been activated by a T helper cell, you can divide and become a plasma cell or a memory cell. Now, first off, let's focus on what these plasma cells do. So the plasma cells secrete these really cool little Y-shaped things called antibodies, and the antibodies attach themselves to the invaders. So that's pretty good. Now, the other thing you've got are the memory cells. So the memory cells, coming from up the top there, they don't do anything to begin with. Their whole job is to activate if the same antigen comes back into play. So if the same pathogen is there, that's what they'll do. And then this here is a whole nother lesson that we're going to talk about, those antibodies and what they do. So the antibodies bind to the antigens wherever they are, and they can clump them together, and that's great. And then you get all of these eta cells. Now, I'm hoping you understand that an eta cell is a macrophage or a phagocyte. Now, if you're getting confused about macrophages and phagocytes, Just remember that phagocytes is any eating cell and a macrophage is a type of phagocyte. So I'm just going to write that down for you. So macrophage, if I can, is a type of phagocyte, because phago means eat, site means cell, so a phagocyte is any cell that does eating. A macrophage is a big eating cell. So that's why I've chosen to say this. But it could be a macrophage, it could be a neutrophil, mostly it's macrophages, or it could even be a dendritic cell. Okay, but all of these get eaten. You can just all call them phagocytes. There's another way of looking at this. So here is your APC. So this is your APC. It presents the antigen to a T helper cell. The T helper cell activates a B cell, and then it does two things. So this is the activation is by cytokines. So remember that a cytokine is just a signaling molecule in the immune system. All the way back from that one. We're linking them together. So a cytokine is just a signaling molecule in the immune system, and it does two things. We're going to split off and make plasma cells, B plasma cells, and we're going to split off and make B memory cells. The plasma cells, its job is to create antibodies, and then the antibodies are going to neutralize things using the next video that I'm going to go through then. So recapping it, think about what are the main leukocytes, what is the function of these leukocytes, give a brief account of how they would destroy an infective agent, which we're going to come back to next lesson, and how does this differ from the cell-mediated response. So I want you to pause, and I want you to come back and see how we go on these. Amazing, all of the answers appeared. So the first thing is, what are the main leukocytes? I need to have three: a T helper cell, a B plasma cell, and a B memory cell, even though I've got oh no, I just lost all my answers. Oh, well, that's okay. You guys can go back and pause and have a look at those answers. And that's the end of my video. No, so I've gone through the function. You can go back and have a look at the function. This brief account on how they're going to work is going to be next lesson, and then next video. And how does it differ? That's all back there in those answers as well. But that's just sum up on the bottom here. So this is on the top of page 15. What are the difference between a plasma cell and a memory B cell or a B memory cell? It doesn't matter what order you say it in. So the plasma cells are involved in the primary immune response, which we'll come back to. They secrete antibodies, but they aren't long-lived, so they only survive for a few days. You get lots and lots of them, and then they die. Now, the response is quite slow, and usually you get sick before, like you feel symptoms before the pathogen is killed. Now, the memory B cells, they come back when you have a secondary exposure. So secondary immune response, so this is when you are reinfected. So these are the things that we want. When we're talking about coronavirus, and if you had the coronavirus, you would have developed memory B cells. So it means if you get re-exposed to the coronavirus, you should be able to get these guys into action. So they circulate in your body and your tissue fluid, and when they get to the antigen, when the antigen comes back again, they divide really rapidly. So they go bang, 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 divide really rapidly, and because it's so fast, you're unlikely to feel symptoms. So that's why the benefit of a memory B cell comes into play, and this is exactly how a vaccine works. But we've got a whole nother lesson on vaccines to talk about that. But that is the end of the adaptive immune response, the humoral response.